So in section 5, we're going to talk more about the humoral immune response. Just as a reminder, this is one of the branches of the adaptive immune system. The other one was the cellular immune response. And the function of the humoral immune response was really to target extracellular pathogens by producing antibodies. And the antibodies are just going to bind to the antigens and activate them and mark them for destruction by another cell that comes in. The antibodies themselves aren't actually going to get rid of the pathogen or the foreign debris. So we talked a little bit about naive cells. So if we have a naive B or T lymphocyte, that just means that it hasn't encountered its antigen yet. So that's why we were sending the lymphocytes to the secondary lymphoid organs like our liver and our spleen so that hopefully they can be exposed to a specific antigen. When the naive B cell or B lymphocyte is exposed to its specific antigen, it's going to bind it using its receptor. And a B lymphocyte's receptor is an antibody itself that it has on its surface. Once it recognizes this antigen, it's going to basically bring it in and internalize it, process it, and then chop it up and put those little fragments on its surface using that MHC complex that we talked about previously. So it's going to kind of be in the groove of the MHC complex on the surface of a B cell. So B cells actually, like we talked about, going to act as an antigen presenting cell to show another cell, which we'll talk about later on, called a helper T cell, that, hey, this is what it looks like and it's not supposed to be here. So once we have our B lymphocyte binding to its specific antigen, we then consider it to be activated. And once it's activated, it's going to start producing clones of itself and differentiate into plasma cells. So our plasma cells are going to be the ones that are actually producing the antibodies. We also talked about how the adaptive immune response has memory. So along with the plasma cells, we're also going to produce a couple of memory B cells, which are going to allow us to produce a much faster response if we encounter the same antigen again. So that's kind of what's shown to you in this image over here. We have our activated B cell. Once it has been exposed and bound to its very specific antigen, the majority of cells that are going to be produced are going to be plasma cells. These are the effector cells because they're going to secrete antibodies. And then we'll also produce a couple of memory B cells that are just going to persist in our blood, again, just in case we were to ever encounter that antigen again. As I mentioned, the antibodies that are going to be circulating in our blood and our lymph, they're just going to bind to free antigens and then mark them for destruction or inactivate them. So this is just showing you a little bit more about the memory cells and the importance of memory when we're talking about the adaptive immune system. So when we're talking about immunological memory, we have primary and secondary immune responses. And what we're talking about, primary means the first exposure to that antigen, and secondary immune response would be subsequent exposures. So the second, third, or potentially fourth or more. When we're talking about the primary immune response, it's a lot slower, so that's why we make those memory cells just in case we encounter it again. It can take anywhere from 5 to 10 days before we have significant antibody levels appear in our blood. Most of the cells that are going to be involved are going to be our naive B and T cells, and we'll talk about specific antibodies later. But IgM is going to be the first immunoglobulin that's produced, and then later we'll have some IgG. And when we're going to make those memory B cells so that we can have them ready to go just in case we have a future response. Strength-wise, relatively short-lived, the primary immune response, and relatively weak. We also have the secondary immune response, which is going to occur if we have subsequent exposure to the same antigen. Because we have those memory B cells available, it's going to be a much faster response. 1 to 3 days, we could have our antibodies ready. Really rapid and high production of these antigens. antibodies, and we're going to produce a variety of different types of antibodies depending on the antigen. So those memory cells are going to be reactivated, and we can even cause further proliferation. Strength-wise, much stronger, much more specific, and a much longer lasting response. And that's what's shown to you on the graph over here. So here's our primary immune response here. We can see over here is basically the level of antibodies we have in our plasma. Primary immune response, we're not going to have a huge amount of antibodies being produced. If we were to be exposed to the same antigen again, we can see we produce a much higher number of antibodies against that antigen. And if we look at the time, this has a much steeper slope in order to get that peak of antigen levels in our bloodstream. So much faster and much more robust if we're talking about a second